Okay, in this video, we're going to talk about decisions that we can make when hypothesis testing. And sometimes we're going to make the correct decision, and sometimes we're going to make an error. So another way to frame this or think about this is errors in hypothesis testing. So let's first consider what exists in nature. And so I have it labeled up here, the truth. And one possibility is that the null hypothesis is true. And so you know from our other discussions in class and videos that that means that the groups belong to the same population. Or another way to think about this is that whatever you've done in your experiment, the independent variable does not affect the dependent variable, right? And so that's represented here in this column. The other possibility, right, is that the null hypothesis is false, meaning the groups don't belong to the same population. Whatever you did to the independent variable caused a change in the dependent variable, or right, you could have some sort of confounding variable, but the groups are unequal now; they're not the same. There's some treatment effects. So that's what exists in nature, and what we're doing as a researcher is we're trying to discover this truth, whatever it is, the null hypothesis being true or false, and we're testing the null hypothesis. So over here, you can see the two options for our experimenter's decision. We gather the data, we run our statistical analysis, and we're either going to reject the null hypothesis or we're going to accept the null hypothesis, right? So we do one of those two things. And so if we reject the null hypothesis when it's true, well, that's a type 1 error, right? We've, in effect, said that there's a difference that doesn't exist. It's not true. So this is represented by alpha, which is the probability of a type 1 error. And we'll see that graphically represented here in just a second. And then we'll talk about, in a separate video, we'll talk about how to sort of change, maximize our correct decisions, and change around these probabilities. Now, the other thing that we could do is we could accept the null hypothesis when it's true. Well, this would be a correct decision, and that's represented by 1 minus alpha. And the reason it's represented by 1 minus alpha is, remember, this is going to fall under one entire frequency distribution, right? So if you have one area which is alpha, everything else sums to 1, so 1 minus alpha is the other area. Now, the other thing that could happen is we could reject the null hypothesis when the null hypothesis is false and should be rejected. Well, this is a correct decision. And this is represented by 1 minus beta. It's also called power, statistical power. It's something that we're really interested in as researchers, right? Because we're setting up our experiment, we sort of expect to have reason to believe that our independent variable is going to do something to the dependent variable. So this is the reason why we're in this research endeavor to begin with. Now, the other thing that could happen is we could accept the null hypothesis when indeed it's false, and we should be rejecting it. That means we're sort of missing an effect, right? And this is represented by beta, probability of a type 2 error. Now, beta belongs to a different distribution, so beta forms an area. 1 minus beta is the other part of this cell, because again, it sums to 1. It's kind of hard to wrap your brain around. It's a little bit better, a little bit easier when we see them graphically represented. We can sort of see that they're two overlapping distributions, but they're different, separate distributions. So alpha and beta are related to one another, but they're not mathematically related to one another. They're inversely related. So as you increase the probability of one, you're going to decrease the probability of another. And that's something we'll explore more in videos and in class. So let's look at this. Let's look at this graphically. So you'll get a couple of distributions here in the handouts, and I'm going to focus on this one right here, and I'm going to draw on a little bit. So this capitalizes on our previous discussion, video discussion, about the sampling distribution, right? So if this is a sampling distribution of our group means drawn from the same population, so we'll assume that this is the null hypothesis sampling distribution, okay? And remember that we're going to have most of those samples will fall within the 95% confidence intervals. And down here I have the x-axis here was generated. This is just generated in a computer program that creates a Gaussian distribution, but those differences don't mean anything, so that's why I deleted them there. But so you can ignore the values down there. It'll be more important later when we talk about changes in it, so you can sort of see how they're changing, but they're meaningless other than that that's what they use to generate this particular distribution. And so when we look at this, this would be if our group mean differences fall in here, and all of this data belongs to the null hypothesis, right? This would be the area that's 1 minus, and I can't do alpha on the keyboard, so it's 1 minus alpha, right? It's a correct decision, right? This is when we're going to retain the null hypothesis when we should. They belong to the null hypothesis, right? And if our differences in our group means fall out here to the left, what we're going to do is we're going to reject the null, right? We're going to do that if it falls out here into this region and anything out here to the left, because our differences are greater than what we would expect by sampling variation. Same is true out here to the right. We're also going to reject the null if our test statistic is out here, out to the right. Okay, so in many cases that's going to be a correct decision, but for this area under here, these sampling distribution means actually belong to the null hypothesis, or when they belong to the same population, right? There's a very small number of cases out here too. Very small number of cases that belong to that, right? And so actually we quantify this because we set alpha, right? We set the alpha level at 0.05, right? So that means that actually out here it's 0.025 in this tail, and there's 0.025 in that tail, right? And this is the probability of a type 1 error, right? So this is when we reject the null hypothesis, but in reality, the null hypothesis is true. So let me see if I can represent that real quick. So in reality, the null hypothesis is true because if it belongs to this area down here, it was sampled from the same population. Our independent variable didn't do anything, and the null hypothesis is true, but yet we're rejecting it, right? So that's the area that's represented, this is the area that represents statistical error. We made a false positive identification. Okay, so that's sort of the basics. What we're going to do in the next video is we'll overlap our other distribution, because the reality is here you have multiple populations, other other than the null hypothesis population. And we're going to explore that as well as talk about how you can maximize correct decisions.